Hello and welcome to another special bonus episode of Damn Interesting Week. This bonus episode is all about birth and birthdays, and that's because this Saturday, November 15th, is our own 6th birthday of doing this podcast. In those 6 years, we have said the phrase next link 1,458 times, which, if you add it to the 241st links, gives us a total of 1,698 articles, which I gotta admit was a slightly frustrating number, but on exactly 4 occasions in the last 6 years, we have done a twofer, which is to say we had one discussion that actually referenced 2 different but related links. That pushes us over the top for a total of 1,702 articles. Also, coincidentally, last week was my 45th birthday, which means that if we keep this up, we can just manage to hit our 3,000th article before my 50th. Hopefully you're up for that, because we definitely are. And if you want to get us a birthday present for this momentous occasion, there's 2 things you can do. Number 1, we have started releasing episodes a day early for our Patreon subscribers. If that appeals to you, we'd love to add you to the list, because we remain, as always, fully committed to never running advertisements on our website or any of the Damn Interesting family of podcasts. The second thing you can do, and this one's completely free, is you can tell your friends about us. Leave us a review. Tell everyone in your social media feed that we are hilarious or clever or just very sexy. I'm open to all compliments. Whatever avenue you choose, it really does make a difference, and we don't want to be anybody's best-kept secret. Thank you again for spending 6 wonderful years with us. I'm going to go blow out some candles, and in the meantime, we hope you enjoyed this look back through the archives. My name is Jennifer Lee Noonan.
I'm Angela Epley.
I'm Spurr-Chen.
I'm Bradley Calhoun.
And these were some damn interesting weeks.
First Link.
This article comes to us from TheGuardian.com. It's titled, All South Koreans to Become Younger as Traditional Age System Scrapped.
Whoa!
Excellent.
Lucky.
I have always thought this was so stupid. And I don't want to be like, cultures do things differently. But oh my God, what an incredibly stupid system they've had. Anyway, go ahead and explain it.
So with that preface,
Yes.
South Korea is to scrap its traditional method of counting ages and adopt the international standard, a change that could knock 1 or 2 years off people's ages on official documents, but could take time to seep into daily life. South Koreans are deemed to be a year old when they're born, and a year is added every January 1st. The unusual and increasingly unpopular custom means a baby born on New Year's Eve becomes 2 years old as soon as the clock strikes midnight.
Uh-huh.
The complications do not end there. A separate system exists for calculating the age of men entering national service and the legal age to drink alcohol and smoke. In those cases, a person's age is calculated from 0 at birth and a year is added on New Year's Day. The tradition has attracted criticism from politicians who believe it makes South Korea appear behind the times. The president, Yoon Suk-yeol, has criticized the multiple methods for calculating ages as a drain on resources. The confusion should end in June, at least on official documents, when laws stipulating the use of only the international method of counting ages take effect. Jeong Da-yoon, a 29-year-old office worker, said she welcomed the change since she always had to think twice when asked her age when overseas. She said, I remember foreigners looking at me with puzzlement because it took me so long to come back with an answer. The system's origins are unclear. One theory is that turning 1 year old at birth takes into account time spent in the womb, with 9 months rounded up to 12. Others link it to an ancient numerical system that did not have the concept of 0. Explanations for the extra year added on January 1st are more complicated. Some experts point to the theory that ancient Koreans placed their year of birth within the Chinese 60-year calendar cycle, but at a time when there were no regular calendars, tended to ignore the day of their birth and simply added on a whole year on the first day of the lunar calendar. The extra year on January 1st became commonplace as more South Koreans began observing the Western calendar. While some people are expected to continue using their Korean age in daily life, others said they were delighted by the prospect of turning back the clock. One tweeted, I'm getting 2 years younger. I'm so happy. I turned 2 years old so soon after I was born, as I was born in December. Finally, I'm about to get my real age back.
Yeah, I mean, I imagine some people are resentful because it's like, oh, you're allowed to drink. Oh, psych, you're not allowed to drink again. But yeah, for the most part, it absolutely boggles my mind that this system has lasted as long as it has. And as someone who cares about numbers, it infuriates me. So I'm really glad. This is good news. I'm happy.
Has this ever affected you directly?
No, there's absolutely no reason I should feel this worked up about it. When I first learned about it, I don't know, it was probably 10 years ago, I read something somewhere that they're like, oh, this is the system in South Korea. And I'm like, this has got to be a joke. Nobody does this. And they were just like, no, this is how we calculate age. And I was like, you can't have a functioning society where a 2-day-old baby is in class with 2-year-olds. It doesn't make any sense. Anyway.
Dogs and cats living together.
Exactly.
Total anarchy.
So, good. I have one less thing to be angry about now in the world.
Yes. Next link.
Next link.
Okay, some promising, happier news. This is from the new Atlas. Promising male contraceptive pill works in 30 minutes and wears off in a day.
Ooh.
Hey.
Wow.
Yeah. So I think it's fair to say that for decades, women have shouldered much, if not all, the responsibility for contraceptives.
Mm-hmm.
And the past attempts we had were ineffective. They either lasted too long, took too long to kick in, and just had way too many side effects. So a team at Weill Cornell Medicine has a pill that seems to solve these issues. In mice, let me be clear.
Yeah.
It works by targeting a protein called soluble adenylyl cyclase, or SAC, which is vital for sperm function. That makes the drug a SAC inhibitor.
Oh, no.
These scientists and their punny names, right? Yeah. So they gave male mice a single dose of the SAC inhibitor called TDI 11861. I'm not sure if I get that pun, though. And then after 52 mating attempts, not a single mouse became pregnant compared to about a third of the control group.
Wow.
Apparently, it works rather quickly, within 30 to 60 minutes, remains 100% effective for up to 2.5 hours. By the third hour, some of the sperm regain their motility.
So it basically is like a tiny little internal stun gun for sperm. It's like, go to
They
sleep.
just can't swim. Yeah.
Yeah.
Right. And then after 24 hours, they were all back.
Yeah, if it starts to wear off in 3 hours, you're starting to cut it close. Like the ideal would be about 6 to 8 hours.
Right, or 24,
Okay.
yeah.
Well, sure. I'm just saying, like, you're going out to the clubs or whatever. You take it at 9 p.m. By midnight, you're in trouble again. That doesn't get you to the 2 a.m. last call. So
There is a lot of variable in terms of timing, though. I mean, that struck me as well. Like, it seems to be a great way to develop some, like, weird anxiety about setting alarms to get the timing exactly right.
Right, this has
despite
to happen right
raging
now.
Hormones. Yeah.
yeah.
I'm sure in time they'll figure out ways to extend that
Sure.
lifespan out a little bit.
Yeah, give it like a slow dissolve coating where you're taking like multiple doses over the, they can make that work.
The slow release.
Yeah.
There are somewhat alternatives to this right now that are temporary, but they're not great. They can take weeks to work or to regain fertility, and it involves, let's just say, a rather invasive procedure.
Well, yeah, invasive procedure is no good. But like a woman's birth control pills, they basically take a month to kick in reliably because you got to go through a whole cycle.
Mm-hmm.
And if you're going to stop taking them, you're looking at 3 to 4 weeks before you're back on your own normal cycle. So, that feels like a weak complaint if it takes
Agreed.
a
And
while to kick in.
Right. I would also argue that the pill is somewhat invasive. It's just not,
Oh, yeah.
it's a hormonal invasion.
Well, yeah. And I read a thing once that was talking about, you said they had canceled some early attempts at male contraception because they had too many side effects. And when you actually looked at it, the side effects the men were experiencing were basically identical to side effects that many women get from birth control pills. But they were just like, oh, no, don't let them have headaches. That's so sad. And so they just said no.
Not headaches for men.
Yeah,
No.
yeah, exactly.
No. So, no testing in humans yet, but we'll see if it carries across, which it does, but we will probably legislate that right away as well.
Yeah, but speaking of conspiracy theories, this is where somebody comes up and says they're putting it in the water. And
Oh, boy.
Turning the frogs gay.
there you go.
Yep,
There you
yep.
go.
Mhm. Next link.
Next link. All right, let's stick with biology because interestingengineering.com lives up to its URL with this article. Every sperm for itself is a myth. new research finds.
Are they like working together? Like it's conspiracy?
Well, more like cooperation because, listen, it is a survival trait. All right, we're going to be using some adult language here, but this is just so hugely fascinating that the takeaway overrides my 12-year-old inner giggle boy. Okay, so we've had this long-held view of the singular Michael Phelps-esque swimming sperm, right? It's often been represented as individuals racing against each other in competition to fertilize an egg. But the problem with this stems from looking in the wrong place. In other words, we get this information from a microscope, and sperm don't really do a whole lot underneath a microscope because most sperm action happens inside the female reproductive system, like it's supposed to, right? Because of this, we've had several persistent misconceptions, like the notion that sperm competition is a constant aspect of reproduction. Now, this new research proves that sperm embrace teamwork. That's my Oprah voice, because this is a big deal, y'all. All right. Chi-Kwan Tung, a physicist at North Carolina Agricultural and Technical State University, and his colleagues noticed this clustering in their laboratory in a previous study they had completed in 2017. So from this study, the group knew that bull sperm, which is similar to human sperm, formed clusters, but also those clusters couldn't swim faster than the individuals. So they didn't think there was any, apparent advantage as to why the clustering was happening. Tung explains in a statement to New Scientist, quote, "In biology, when cells and structures do something, they should probably get something out of it." So that became the question we were asking ourselves. So to answer that enigma, the researchers put 100 million fresh bull sperm into a silicone tube filled with fluid that mimicked the cervical and uterine mucus of cows. According to Tung, this fluid, and I'm sorry, the researcher's name is Tung, so I apologize for this sentence. According to Tung, the fluid had the consistency of melted cheese. Okay, we're on.
Oh, okay.
Wow, I really am just sitting in those last couple sentences.
I'm doing my best, y'all. I'm trying to get through this in a straight face. So they've got this tube, it's full of the stuff, and then the scientists use a syringe pump to create 2 speeds of flow. And what they found was that the clustered sperm swam in a straighter line than the individual sperm where there was no flow. So they also found that individual sperm could not move upstream in the intermediate flow, but the clusters could. And when the flow was high, the clustered sperm were much more effective at pushing through the approaching current than the individual sperm, which typically got carried away by the heavy stream. So think about cyclists that ride together. You've got that like downdraft or whatever. It's a similar property here.
Well, it's like birds flying in a V. Like the one at the head is taking the hardest, but the rest are kind of getting caught in that backdraft and they're able to just coast along a little easier.
And even more importantly with that bird metaphor, the one in the front doesn't stay in the front the whole time. Right? They take turns, they rotate. And that's exactly what the team found in this particular instance as well. There was never a single leader sperm. There's no alpha beta situation happening here, okay? Rather, the groups were highly dynamic, with sperm frequently entering and exiting their cluster and moving around with them.
Well, and it makes sense because, I mean, yes, you're different sperm, but you're all the same genes, basically.
Exactly.
And so it's to your advantage, theoretically, if they had brains and could think about it, to all share the effort and make sure somebody gets there, even if it's not you.
And if we are going to be talking about sort of the natural selection of this method, then the cooperative sperm are more likely to succeed than the sociopathic sperm. Not to say that those don't come in one or the other, or maybe like your whole cache is sociopathic, and so it doesn't matter if A or B goes in there, but just trying to change your worldview a little bit here. We got to live on this planet together. The sooner we learn to love that, the better. Didn't mean to make that rhyme. Okay, next link.
Next link.
Well, we'll stay firmly in the animal world, though this is more about their life than their tasty, tasty death. This article comes to us from Gizmodo.com. It's titled, 2 California Condors Had Virgin Births.
Wow.
So in a truly double-take inducing finding, geneticists recently surveyed a population of critically endangered California condors and found that 2 of the birds reproduced without actually mating. This phenomenon, called parthenogenesis, which is a nice callback to Athena springing from Zeus's head, happens occasionally in reptiles and fishes, but rarely in birds.
Wow.
The scientists stumbled across the discovery when going back through the genetic records of the condors from the mid-2000s. It's a remarkably improbable occurrence, said Oliver Ryder, a geneticist at the Wildlife Alliance at the San Diego Zoo and co-lead author of the paper, in a phone call with Gizmodo. In their lifetimes, they weren't even recognized to be parthenotes, so we're definitely keeping our eyes out anytime we get a batch of blood samples for testing. Condors are the largest birds in North America, with a nearly 10-foot wingspan and weighing in at over 20 pounds in some cases. With an attractive ruff of black pointing feathers around their neck, they often look like they're going to a fancy Halloween party. This discovery, a total goosebump moment, according to Ryder, occurred when the team was taking stock of the historic bird population to maintain the genetic health of the condors, because when small populations of animals are being bred, there's a risk of inbreeding or genetic bottlenecks which can make the animals less robust. The team found that one male chick in the population and then another did not share genetic information with the male condors presumed to be their fathers. The researchers then checked 487 other male birds for possible parentage, but none of them had bequeathed their genetics to the two chicks. Not even close, Ryder said. Both chicks were also homozygous for their mother's genetics at 21 marking points along their genetic code, and the team found that none of the other male birds qualified to be fathers of either chick based on differences in their DNA. Therefore, the team determined the young birds were solely the makings of their mothers. Though the mothers themselves were not virgins, they had had 11 and 23 chicks respectively before these two special cases. You know, if that matters to you, I guess.
Right.
Previously, there have been documented cases in finches, pigeons, turkeys, and chickens, but never condors, and the finding has heightened significance given the condor's endangered status.
Yeah. I mean, this feels like divine intervention. Did they name her Madonna?
Yeah. We'll get to that. So in 1982, there were only 22 birds alive. Established breeding programs have since boosted that number to over 500, but the animals remain at the brink of extinction. The two chicks would be capable of reproduction, at least in theory. Unfortunately, they are both dead.
What?
Yeah. Studbook number 260 died at the Condor Sanctuary at Big Sur in 2003, and Studbook number 517 died at the Oregon Zoo in 2017. So it's not like they, died immediately. They had some lives for a while, so that's nice. They're just not around anymore, unfortunately. But anyways, so turkey parthenotes can reproduce, so the same likely applies for their carrion-loving cousins. That said, it's not like parthenogenesis can save a species from extinction. It's an extremely rare phenomenon, and the rhyme or reason for its occurrence in the condors remains an enigma. Ryder said, "We only now have the genetic tools to look at this in detail. Previously, parthenogenesis was really identified by seeing females who weren't housed with males have offspring. But now we know the condor can have offspring while being housed with males, and it begs the question, is this going on more than we know?" So while the research team will keep plugging away at conservation, Ryder said they've likely deduced all they can from these two unexpected cases, but they'll keep an eye out for more of these virgin births in the future.
I mean, it's fascinating to me that, because my understanding of parthenogenesis was that you're basically creating a clone of yourself. But that can't be the case if it's a male bird being born to a female bird. But then I also know, bird genetics are weird. Like, they don't have X and Y chromosomes. They have a whole separate system, which I think is like Z's and W's. I don't remember. You can't ask me any more information because that's all I have. But but it still seems very strange to me that clearly this bird had some level of genetic difference from its mother. Where did those genes come from? And, maybe it was all mutations.
How are they stored?
Yeah. Maybe it just, maybe that's why it died, because it was a genetic freak. But
yeah, potentially. It was published in the Journal of Heredity. If anyone would like to look it up, I imagine there might be some information there. Oh, they've actually got the full article just publicly available. So if you are a genetically minded person and you're curious, you can go check that out.
And tell us if there's any chance of it happening in humans.
Yeah.
Because that changes a lot.
Yeah, it's a complication for sure. Next link.
Next link.
All right, well, this next link comes from Discover Magazine by Riley Black. It's called, "Why Did Our Mammal Ancestors Stop Laying Eggs?"
Oh, good question.
It is a good
Yeah.
question. We should start by pointing out the obvious. Some mammals do still lay eggs, namely the duck-billed platypus and the spiny echidna. And these creatures belong to an archaic mammal group known as monotremes, which split off evolutionarily from the rest of us more than 100 million years ago. So it is a given that we all used to lay eggs. It's just a question of why did we stop? And to figure out when the rest of Mammalia stopped laying eggs, we have to go back over 300 million years ago, when the first mammal ancestors split off from the shared reptile ancestors.
Okay.
And this new branch of creatures were known as synapsids. And at that point, we know for sure they still all laid eggs, and they continue to do so for millions of years. So here's where we get to a big gray area in the fossil record. There's only a few key data points. For example, in 2018, paleontologists Ava Hoffman and Tim Rowe announced that they had found an entire clutch, meaning a fossilized mother and a whole group of babies, of a proto-mammal called Kayentatherium.
Ooh.
This was a weasel-like proto-mammal that we had fossils of before, we knew, but we had never found one with all the babies all together in the litter.
Ooh.
And it lived during the Jurassic about 185 million years ago, and Hoffman and Rowe concluded that the Kayentatherium must have laid eggs because the sheer size of the litter was too big for live births to have been possible. Unfortunately, they have to rely on deductions like these because egg-laying mammals, including the platypus and the echidna, generally lay soft-shelled eggs, which are more likely to decompose and not get fossilized. They've really never found a bunch of good fossilized eggs to figure out at what point did this stop. Another key place paleontologists look for clues is pelvis size. As the pelvis of a particular species gets smaller, it gets more and more likely that that animal is giving birth to live young rather than laying eggs. And this didn't actually make sense to me because I think of lots of small eggs versus one big baby, right?
Right. Yeah, wait, wouldn't a smaller pelvis make live birth more difficult?
Right. Well, see, this is what I thought. But you got to think about like a chicken laying an egg. It lays exactly one egg at a time, right? So if a human woman were laying an egg instead of a baby, you would have, imagine like the entire baby all balled up. Whereas live young can kind of like slither out lengthwise, and you only have to and you only have to worry about the size of the head getting through the birth canal. So actually giving birth to live young, it's easier. You can kind of squish them out as opposed to like forcing a giant ball of an egg out. Deep breaths.
I'm good. I'm good. Yeah.
And, and if you really want to get specific with it, basically the placenta is sort of like an eggshell. Like that's where the shift happened, where we do have this sort of shell around the baby, but then the water breaks, the placenta opens, and the baby comes out without the entire placenta going all in one go. And they said, overall, scientists say there's nothing inherently superior about live birth. It just seems to kind of be another way of doing things that's sometimes useful and sometimes not. And in fact, we can't even be certain that marsupials and mammals share a common live birth having ancestor, because live birth has been shown to evolve independently over 100 times in the fossil record, including many reptile species that actually have live births, including boa snakes and blue-tongued skinks. The boa snake and the blue-tongued skink share a common egg-laying ancestor. They split off, they both were still laying eggs, and then each one independently shifted to live birth. So, unfortunately, the end result is we don't know. It's really very, very fuzzy, and until we find that, holy grail of a clutch of eggs from particular mammals along the evolutionary path, we're just kind of not going to know. I will close on a fun fact from the article that is only loosely related but was incredibly adorable. Baby monotremes, such as baby platypuses, are called puggles.
Oh.
So yeah, if you ever see a baby platypus, it's not a platypus, it's a puggle.
Well, dadgummit. That is precious.
So that's some cuteness to kind of a chaser for the horrible.
For the slithering that will never escape my mind. That word choice is that did me in.
Next link.
Next link.
This article comes to us from livescience.com, and it's titled, Lab-Made Mouse Embryos Grew Brains and Beating Hearts Just Like the Real Thing.
Whoa.
Aw.
Yeah. So the lab-made embryos crafted without any eggs or sperm and incubated in a device that resembles a fast-spinning Ferris wheel full of tiny glass vials survived for 8.5 days.
Oh.
And that's nearly half the length of a typical mouse pregnancy.
Oh, wow.
In that time, A yolk sac developed around the embryos to supply nutrition, and the embryos themselves developed digestive tracts, neural tubes, beating hearts, and brains with well-defined subsections, including the forebrain and midbrain, all of which was reported in a study published Thursday in the journal Nature. Senior study author Magdalena Zernicka-Goetz, a developmental and stem cell biologist, said in a statement, "This has been the dream of our community for years and a major focus of our work for a decade, and finally, we've done it." The new work produced very similar results as an earlier study published August 1st in the journal Cell, which was led by Jacob Hanna, an embryonic stem cell biologist at the Weizmann Institute of Science in Israel and co-author of the new Nature paper. Although the two recent studies produced similar embryos, the experiments started out slightly differently. In the Cell study, the researchers started by coaxing mouse stem cells into a naive state from which they could morph into any cell type, such as heart, brain, or gut cells.
Ooh.
From there, the team divided these naive cells into 3 groups. In one group, they switched on genes to form the placenta, and in another, they switched on genes to make the yolk sac. The last group they left alone to develop into embryos. Zernicka-Goetz's research group, on the other hand, began with 3 mouse stem cell types, rather than starting with only naive cells. One type of stem cell gave rise to the embryo, while the other 2 morphed into the placental tissues and yolk sac. Throughout the experiment, they observed how these 3 stem cell types interacted, exchanging chemical messages and physically butting up against each other in the glass vials. Studying such exchanges could give hints as to how the earliest stages of embryonic development unfold in humans and what happens when things go awry. In both the Cell and Nature studies, the resulting synthetic embryos closely resembled natural embryos, albeit with some slight differences and defects in how the tissues self-organized. However, in both experiments, a very low proportion of the stem cells actually gave rise to embryos, suggesting that the efficiency of both systems could be improved. In addition, neither set of synthetic embryos survived to the 9th day of development, an obstacle that would need to be overcome in follow-up studies.
Yeah, they died. That's an obstacle.
Yeah, a pretty big problem, right? With the generation of life. And the last line of the article ends, "The research also raises ethical questions about how and if such technology might be applied to human cells in the future."
Yep.
Does it? Does it maybe? Like,
Yeah, just a little bit.
I mean,
That concession to the obvious was a really nice way to end, especially given all the very careful treading around. Yeah, they didn't make it.
Yeah.
Yeah.
Well, we did recently have the first full-grown calf birth from a artificial womb. That happened a few
Ooh.
months ago.
mhm.
So I think they started with like a real animal, and put it in an artificial womb. But you've
Right.
got both of those technologies progressing, and
Put them together, man. It's like peanut butter and jelly.
yeah, delicious, yummy.
Tanks of growing humans just ready to be organ harvested for our. I mean,
You're
no.
way ahead of us,
We'll,
Jen.
we'll only use this for good. Why would I suspect otherwise? Like,
Yeah, absolutely. We have a very good track record of that with our science.
yeah,
Gosh, even
absolutely.
just the phrase synthetic embryo, that's wild, y'all.
Yeah, it really, really is. Next link. I'm just overwhelmed. Let's just move on.
Yeah.
Next link. Okay, from technologyreview.com, we've got an update for those of you making end of life planning changes. The holidays are such a great time to get together with the family, but real talk, you better know what the wants of your loved ones are, because it's going to save a lot of grief if you're trying to guess that after the fact. But one factor we all need to take into account now is telling partners or parents whether you want your eggs, sperm, or embryos to be used after you've died.
Oh,
So we start with an example. Peter Zhu, he was only 19 when he died following a skiing accident in New York. His donor card made it clear he wanted to donate his organs, but his parents wanted to collect his sperm too. They told a court that they wanted to keep the possibility of using the sperm to eventually have children that would be genetically related to Peter. The court approved their wishes, and Peter's sperm was retrieved from his body and stored in a local sperm bank. We have the technology to use sperm and potentially eggs from dead people to make embryos and eventually babies. And there are millions of eggs and embryos and even more sperm in storage, ready to be used. So when the person who provided those cells dies, like Peter, who gets to decide what to do with them? That was the question raised at an online event held by the Progress Educational Trust, which is a UK charity for people with infertility and genetic conditions. The panel included a clinician and 2 lawyers who addressed plenty of tricky questions, but provided very few concrete answers. In theory, the decision should be made by the person who provides the eggs, sperm, or embryos. And in some cases, the person's wishes might be quite clear. Someone who, for example, might be trying for a baby with their partner may store their sex cells or embryos and sign a form stating that they are happy for their partner to use these cells if they die. But in other cases, it's a lot less clear. Partners and family members who want to use the cells might have to collect evidence and convince a court the deceased person really did want to have children. And not only that, but that they wanted to continue their family line without necessarily becoming a parent themselves. Robert Gilmore, a family law specialist based in Scotland, said at the event, quote, "The law in this area makes my head hurt." And that's also because the law varies depending on where you are, too. Posthumous reproduction is not allowed in some countries, and in many others, it's just unregulated. In the US, laws obviously vary state by state. Some states will not legally recognize a child conceived after a person's death as that person's offspring, according to the American Society for Reproductive Medicine. And the people who want to use the eggs, sperm, or embryos of dead partners or family members are often painted as selfish, but in James Lawford Davies' experience, that just isn't the case. He's a UK-based solicitor specializing in reproductive and genetic technologies, and he has already been involved in similar cases. Quote, "All of these cases have involved incredibly brave people who have been faced with tragedy, and they all wanted to deliver on the wishes of those who had died. But there's one thing that everybody who has spoken on this topic agrees on. Each case is unique and should be treated individually. It's just really hard to generalize when it comes to this kind of stuff. So in the same way that we think about organ donation, we should all be writing down whether, like, yeah, go for it. Let's see what happens. Or, you know, please don't."
Yeah. I mean, it's tricky because thinking about it, my instinct is like, no, I don't want people making a bunch of babies out of my genetic code. But if it were discovered that my genetic code had some sort of crazy gene that could cure whatever, and then they couldn't get at it because I had gone and signed this thing thinking, oh, I don't want you to make a bunch of babies out of me. Like, I would feel bad. I would want some sort of magical genetic material that happened to be inside me, they found out later, to be usable.
And the thing with legal documents, especially estate planning, is you can really customize the absolute crap out of them. And
that's true.
I know I started this off with like, oh, Thanksgiving holiday table. The article in MIT Technology Review quotes the biologist as saying, "We often tell people that Thanksgiving, when you're sitting with your family, it's a good time to express your wishes. They are very hard conversations to have, but how else are you going to make your wishes known?"
Yeah, and it would definitely liven up the Thanksgiving dinner table, honestly. Like, that would be fun.
You want to wrestle this conversation away from political matters, I promise posthumous reproduction is the way to go.
Excellent, yeah. All right, well, that's going to wrap it up for this bonus episode. We hope you've enjoyed it. Don't forget, if you want to start getting episodes early, you can check us out at patreon.com/damninterestingweek. In the meantime, my name is Jennifer Lee Noonan.
I'm Angela Epley.
I'm Spurr-Chen.
I'm Bradley Calhoun.
And we hope you have a damn interesting week. Bye-bye.